Hi, I'm Gabriel Newcomer. This is my submission for the Breakthrough Junior Challenge. For my video, I'm sharing with you how I made the astonishing discovery that DNA, protein synthesis, and proteins themselves are nothing but a large-scale instance of an extraordinarily simple mathematical model called an L system, and how, by extension, these can all be modeled exquisitely as a fractal. I will first detail the generation process of an L system. Then I will highlight its connection to the ACTG nucleotide base pairs that are present in all DNA. An L system is a type of fractal that's specialized for modeling chaos. It relies on an iterative process that instructs a computer on how to develop a set of instructions, and these instructions do the same, so on and so forth, until you approach the final set of instructions, which is the generation sequence. For example, the computer starts with a list of letters, and on each iteration it replaces every letter with a predefined substitute, which is another list of letters. The final list is the generation sequence, where each letter corresponds to a very small, basic unit of instruction. L systems have been known to be beautiful, both for the emergent complexity based on a very simple generation process, or beautiful for their resemblance to things we know and love like trees and landscapes, but I'm going to add a new layer to this beauty. A layer that may revolutionize the medical field and most fields of biology. Here's where DNA comes into play. It's quite simple. ACTG. Although it is widely known that these four nucleotide base pairs literally instruct protein synthesis, it has never before been modeled as such a simple mathematical model as an L system. Although with this approach it may seem counterintuitive that four very basic units of instruction, ACTG, can generate something as complex as a protein. To address this, I'll point out that the chemical composition of a partial protein changes after each unit of instruction is carried out, and because the composition is different, the protein must react differently to the exact same instructions for every future iteration. The last note I'll make is one on efficiency. This may be an accurate method, but what's to ensure that it would allow for efficient simulations? If an average computer can run 3 to 4.8 billion calculations per second, assuming it has an i5 processor, then simulating these syntheses should be able to rival the actual synthesis time frame of 20 seconds to 3 minutes. This L system approach, with the potential for real-time simulations, is thus a practical foundation for new methods of study. Thank you for considering me for the Breakthrough Junior Challenge. I look forward to hearing from you.